away and can change the bulb when we get going. How many of you have access to the slides in the lecture today? Show of hands. How many of you can see someone else's screen or have notes with you that you can work from? Are you happy to go ahead, seeing as you've made the effort to be here, without me projecting the slides? Hopefully we'll get the slides partway through. So yes, happy to go ahead? Yes?
You have to.
Okay, first of all, I want to particularly address the biomed students that are in the room, which should be the majority of you. There has been a case of exam irregularities shown up, which is particularly affecting this week a chemistry examination, and you've been asked to resit a test on Tuesday, I believe. Is that correct?
Yes.
How much information has Professor Seller given to you about those events? None. He had been posting information on the forums, so we were worried about students cheating and knowing about some of the test answers before they sat the tests. So he has taken it to mean that everyone was cheating, even though we're not sure what evidence he has. So please attempt the test as well as you can. We know you have other tests this week, and we know this has changed the way you will have to revise this week and the implications it has on you. So we will be making formal representation to the chemistry exam boards on your behalf, because we're not sure that this is very fair. It's not fair to anyone. As far as we know, this does not affect the whole unit in chemistry for those doing pharmacology or medicinal chemistry. Is that correct? Any pharmacologists in? Yeah, that. Yeah, I think it only affects the half unit in chemistry.
Everyone has to receive the test. Like everyone has to receive the test, not only the half course unit, like the whole class.
Yeah. The whole unit. Okay.
Yeah.
We want to, if things like this are going to happen, we want it to be fair for all students. Okay? I don't think they know much about how this irregularity came about. They're investigating it. If you've had nothing to do with it, you've got nothing to worry about. The only problem is, of course, it's in your timetable now, and we're busy doing other things. Okay? So we look after that side of things. We'll represent our students and make sure that's reported to the exam board at the end. All I can urge you to do is to get on with it as best you can. Okay? Remember how much it is at the final mark. It's not a lot. Do your very best. Concentrate on the things that are very, very important to you. Okay? If you have problems, come and see Dr. Houston as your year tutor, or myself, okay, who will be representing you at exam boards or whatever. If you're not in a biomedical sciences degree and you have worries, talk to your own year tutor or program tutor. But we're hoping, fingers crossed, you'll be able to get through it and out the other end all right. Okay? If you do have any anxieties, come and see one of us and we'll talk to you about it. Any questions? So you know where we are down the basement of the Rockefeller building B54. So I'm there most of the week. We're going to come and have a chat about what's been going on. So it's not on my door. One of us should be in the office.
Okay, or you can drop either of us an email about this, and again, raise any concerns you have. Again, if this has affected you, make sure that we know about it, and we will try to help and support you as best as we can. Okay, so hopefully I'll have slides as we go through the lecture, so we'll get started and hopefully we'll get some support as we go on. So this was the fourth lecture in a series of 5, and we're going to talk about the regulation of plasma osmolality this morning. So looking at iron concentrations, looking at the balance of those across the kidney. So can everybody see someone else's notes or lean over and see something to follow what I'm talking about? Again, very hard without anything that you can see. So again, shuffle around, move. If someone's got a laptop you can peer over at, then feel free to move around. Okay, so the learning objectives for today were to talk about the establishment of osmotic gradients within the tubular network to look at the transport of sodium and chloride in the loop of Henle. So again, we've done the Bowman's capsule, we've done the proximal tubule, and now we're moving on to the loop structure, the loop of Henle. I want to introduce another hormone, antidiuretic hormone, also referred to as vasopressin, whose actions are to regulate plasma osmolality, so key to our absorption of water within the kidney. Remember, up till now, the osmolality is the same. It's the same as plasma. Nothing has changed. It's still isotonic, so it's still around 290 to 300 milliosmoles per kilogram of water. So we'll look at the actions of antidiuretic hormone and its role in the distal tubules and the collecting ducts to reabsorb water, and look at and summarise the overall function of the kidney. So, the control of body fluids is a very unusual process because we only regulate the intake of water and the expulsion of water at the end. So we regulate our first mechanism, how much water we take in, and we regulate urine production at the end. And in between time, we don't have many mechanisms that can regulate your overall water balance. Okay? It regulates the front end and the back end. So in terms of how much we drink per day, well, the ideal value is to drink about 1.5 litres of water per day. So nothing, we've done a full reset, and nothing, but the room's locked. Okay, thank you. So we take in about 1.5 litres of water a day, and you get about 0.5 litre of water from the foods that you eat per day. And we gain further metabolic water from the breakdown of food products and different reactions such as interchange of bicarbonate and other buffers to gain us just under 2.5 litres of water per day that we take in. In terms of the losses should be around about the same, so 2.4 litres per day in terms of output. And that's balanced by the amount of urine that we produce, how much fluids we lose through the skin in terms of perspiration. We also lose water in terms of respiration. So on a colder day like this, when you breathe out, you can see the water vapour in the air. So we lose water with our respiration, and we also lose some water in feces as well. So normally the two elements are balanced. One day suddenly drink more, then you will urinate more. Some days you haven't drunk as much, you will urinate less, and you will balance it in that respect. Go between hot and cold climates very acutely, and again it will change the amount of water you lose, particularly in perspiration. So you will balance again water input and water output. But it means that the overall... Okay, that was easy.
Yeah, someone pulled the power lead out the sector. I don't know how, but look here.
That's helpful. Thank you very much. So, in a few seconds we'll have slides that you can see. So overall, the normal plasma osmolality is balanced by these two key mechanisms, fluid in and fluid out. So, let's look at the two key aspects of kidney regulation, which vary the amount of urine that's produced. And most of these mechanisms occur between the latter areas of the kidney, and we're going to focus initially on the thick ascending limb of the loop of Henle, then look at the descending limb and the loop, and then finally the distal convoluted tubules and the collecting ducts. So, what the kidney has to do is, remember, at the end of the proximal convoluted tubule, the osmolality is still isotonic. It hasn't changed. So, one thing that we still need to do is transport more of the sodium chloride. So, at the end of the proximal convoluted tubule, we've reabsorbed about 65% of the sodium chloride load within tubular fluids. Again, we don't want to lose too much of those essential ions. So we look for further transport of sodium chloride in the thick ascending ascending limb of the left loop of Henle. And for these mechanisms to occur, we also set up what's called the countercurrent multiplier mechanism within the thin descending limb of the loop of Henle, so within this area. And then we'll come back to the regulation of antidiuretic hormone. So within the ascending limb of the loop of Henle, we've got further transport mechanisms on top of what we've already seen in the proximal convoluted tubule. So here we've got a diagram with tubular fluid here, and we can see we've got a specific transporter here, the sodium 2-chloride potassium transporter, which is going to co-transport sodium, chloride and potassium ions into the cells lining the thick ascending limb of the loop of Henle. Now this is driven as secondary active transport by the sodium pump on the basolateral membrane, again driving a favourable sodium gradient to allow this transporter to be active. So this transporter sits on the apical portion of the membrane. We can also see that a number of cations are drawn across in a pericellular manner between the cells in the thick ascending limb and can be attracted into the vasorector, the specialised tubular capillaries surrounding the loop of Henle. So about a further 20% of the sodium chloride load will be filtered and reabsorbed, and also potassium will be reabsorbed in this manner in the ascending limb of the loop of Henle. Now this transporter occurs in this area, and this is the target for most loop diuretics. So typical ones are fusimide and butanamide, and these are used to balance, so more commonly called water pills. So they are the mechanism whereby you can alter fluid balance and directly change the reabsorption or the loss of water from the diet, and therefore change in blood pressure in a large proportion of the population. So these loop diuretics target the sodium potassium 2-chloride transporter, block its action, and therefore more fluid is left in the urine, and therefore more fluid is lost from the plasma, and it's used to reduce blood pressure. So we've got sodium, chloride and potassium being reabsorbed, and then the next thing we need to think about is we've still got a lot of water there. We've reabsorbed anywhere between 50 and 65% of water in the proximal convoluted tubule. Now we need to absorb further water and further sodium chloride. And this is produced in the loop area of the loop of Henle. So you want to produce altering concentrations of urine so that it meets the normal physiological demands. So sometimes you want more dilute urine to remove more water, sometimes more concentrated urine to preserve water content. So these are variable mechanisms going on all the time. Now, the mechanism name comes from the structure, the way it's set up. So it's called the countercurrent mechanism because there is a countercurrent flow. So you have flow from this end, which would be the proximal convoluted tubule, down through the thin descending limb of the loop of Henle, round the loop and up the ascending limb of the loop of Henle. As fluid flows in, well, it has the same isotonic concentration. We've got about 300 milliosmoles, and it would just flow around the loop and nothing would change. So to change the osmolality, then we need this mechanism to occur. And what happens is that we've got the flow coming round, and this is the interstitial fluid surrounding the loop of Henle. What happens is in the ascending limb, we've got that sodium potassium 2-chloride transporter, which is actively using energy. So again, if you look very closely at these cells, there'll be lots of mitochondria there. They're very energetic cells, and they will be moving sodium, chloride and potassium into the interstitial fluid, which will reduce the ionic concentration of tubular fluid. Now, in this area of the ascending limb, water cannot move. The ions can move by peritubular diffusion and mostly by secondary active transport using the sodium pump. So as ions move, the interstitial fluid becomes more concentrated than the surrounding tubular fluid. So what happens is that we then see that there's a change as fluid comes in from the proximal convoluted tubule and descends down the descending limb of the loop of Henle. And in this area, fluids can move. So in this green highlighted area, ions can move, but water can't. So the concentrations change and you set up a concentration gradient. In the descending limb, ions and fluids can move. And because of the concentration gradient, water is drawn by osmosis from the descending limb of the loop of Henle, so drawn from this area into the surrounding interstitial fluids surrounding the loop of Henle. So water is dragged across, some of the ions can diffuse across, because the descending limb of the loop of Henle is much more permeable than the ascending limb. So let's have a look at what happens. So we've got sodium, potassium and chloride being dragged into the interstitial fluids. As more fluid moves in, then the water will be drawn into the interstitial fluid area. It will be balanced by more ions being dragged in from the ascending limb, and the concentration gradients will start to change. This will progressively keep changing, so more fluid will come in, more ions will be moved in the ascending limb, and water will start to move in the descending limb. And we set up a gradient because as more fluid flows in, it will flow in about 300 milliosmoles. So we set up a gradient as you go down the length of the loop of Henle, getting more concentrated towards the bottom of the loop. So more water moves, it moves down, becomes more concentrated, then the concentrated fluid, the sodium, the potassium and the chloride move into the interstitial fluid. So as this process continues with ions, with water moving, then we set up a greater and greater gradient down the loop of Henle, and this increases through time till we get down to a point that the longest loop of Henle structures can go down to about 1200 milliosmoles per kilogram of water. And the fluid coming out of the ascending limb of the loop of Henle can go down to about 100, 150 milliosmoles, so half the value that it came in from. So there's been significant reabsorption of sodium, chloride, potassium and water through the countercurrent mechanism in the loop of Henle. So let's look at the further regulation of water reabsorption in the collecting ducts. So, as the fluid comes out of the thick ascending limb of the loop of Henle, the osmolality has dropped significantly. And in this area, fluid hasn't moved, but ions have. Now, the permeability around the distal convoluted tubules and the collecting ducts, again, is specific for distal fluids being able to move. So, the distal convoluted tubule does have some permeability, which allows fluid and ion movement to again allow more regulation of the ionic balance. Particularly urea and other elements move within a distal convoluted tubule, and it further allows concentration and water movement in this area. The collecting ducts, however, highlighted here in blue, are impermeable to water and but have some permeability to sodium. And so we can take up sodium, we can lose potassium, again driven by the sodium pump on the basolateral membrane to further increase sodium uptake and potassium loss into the potassium loss in tubular fluid, the sodium reabsorption into the peritubular capillaries. Now, these ducts are some ionic permeability at rest, but they have no permeability to water except under the influence of antidiuretic hormone. So, antidiuretic hormone, also called vasopressin, comes from its original name as it was identified. Vaso meaning acting on the blood vessels, pressin meaning to change blood pressure. And this is released from the posterior pituitary gland. In humans, it's also sometimes referred in some textbooks as arginine vasopressin because it has an arginine at the beginning of the molecule, which is different from other mammalian species, so that dictates it as human vasopressin. And within the kidney, the antidiuretic hormone increases water permeability in the collecting ducts. It also increases the permeability of urea, moving urea from the tubular fluid into the inner medullary region of the collecting duct. And we use urea as a molecule that we know has different permeabilities to its osmolality of some of the other ions, and it can be used to concentrate other fluids by generating chemical gradients within the kidney. Vasopressin also has a role to increase sodium chloride reabsorption in the thick ascending limb of the kidney. So again, driving the ionic gradients so that we don't lose much sodium chloride. So the net effect of antidiuretic hormone is to increase water reabsorption. And I said at the beginning of the kidney lectures, when I ask an essay question on water handling in the kidney, this is usually all I get back, is the regulation of antidiuretic hormone. The regulation by antidiuretic hormone only takes in a third to a quarter of the water reabsorption in the kidney. The majority of the water reabsorption is by osmosis. It's following changes in chemical gradients, most notably in the proximal convoluted tubule, and then in the loop of Henle. And the only regulated portion of water permeability is in the collecting ducts. So when we look at the control of antidiuretic hormone, it is produced in the hypothalamus. It's produced in two key hypothalamic nuclei. So that's clusters of cell bodies of neurons. They're called magnocellular neurons because they've got really large bodies. You can actually see them by eye on a staining slide, even before you put it under the microscope. They are very large neurons. And they sit in two areas called the supraoptic nucleus, and that comes from the name, it sits just outside the optic chiasm. So your optic nerves run along the base of the brain where they cross over, and the pituitary gland sits just under where your optic nerves cross behind your eyes. And the supraoptic nucleus sits just on the outside of where those optic nerves run along the base of the brain. The other location is the paraventricular nucleus. Para meaning around, and it's around the third ventricle, which runs centrally down the centre, or down the midline of the brain. And so from these two key areas, the SON and the PVN, we see that antidiuretic hormone is made in the cell bodies, and then it's transported down to the posterior pituitary. Remember, the posterior pituitary is really elongated nerve endings coming down from the brain and releasing their contents in the area of the posterior pituitary. Unlike the anterior pituitary that makes a lot of hormones in the anterior pituitary itself. So, antidiuretic hormone is transported and released from the site at the posterior pituitary. And the release of antidiuretic hormone is sensitive to very small rises in osmolality. So just a 1% rise in osmolality will change the release of antidiuretic hormone. So as we see here, small changes in plasma osmolality over just 13 milliosmoles per kilo generates a huge change in release of antidiuretic hormone to change the water handling in the kidney. So changes in the concentration of antidiuretic hormone between 0.5 and 5 picograms per ml is enough to account for all of the changes in urine osmolality ranging from 100 to 1,250 milliosmoles per kilogram of water. So huge changes in the concentration of urine depending on the metabolic demands of the body. So, how does antidiuretic hormone act? Well, the hormone binds in the collecting ducts to the cells, and it binds onto an antidiuretic hormone receptor. In terms of its proper name, it's called the V2 receptor, meaning the vasopressin 2 receptor.
Is it a G protein or
It's the G protein coupled receptor, and it activates adenylyl cyclase. You see changes in ATP and cyclic AMP generation within the cell, and it causes phosphorylation of pKa. Now this causes preformed granules that contain aquaporin 2. Now aquaporins are a family of channel proteins which, when activated, become inserted into the apical membrane of the cells and act as water channels to increase permeability of these cells to water. So the water channels are preformed in granules, preformed in vesicles within the cell, and once the right signal is present, they move to the apical membrane and generate water movement across the cells to increase water permeability. Now we can see when this receptor goes wrong, we get a condition called diabetes insipidus. That means, so it's not diabetes mellitus that you've generally heard about. Question? No? It's not the diabetes you've generally heard about, where you've got glucose showing up in the urine. Diabetes insipidus is producing lots of really dilute urine, so people dehydrate very quickly. So these are people that will generally be urinating 4 to 10 litres of water a day. So they will be constantly drinking and are prone to a whole range of muscle and nerve implications because they're losing a lot of essential ions. So they can get tremors, they can get tics, they can get convulsions due to changes in ionic balance. So when people have this receptor go wrong, they produce lots of dilute urine. It can be handled by handling water intake and water balance, and often these people will eat a lot of salt, and that's good for them, even though it's not good in a normal diet. You actually have to replace and encourage these people to limit their water intake and take a lot of salt. So, how does antidiuretic hormone regulate the system? Well, it works in concert with our first mechanisms to balance overall plasma osmolality. So here, if we've got someone who's out in the desert, is very dehydrated, lots of heat, not a lot of water intake, then we would see that plasma osmolality would increase. So our ionic concentration would go up within the plasma, which would activate our osmoreceptors in the brain and cause increased production and release in the PVN to generate more antidiuretic hormone to be released, which would cause the more permeability of the collecting ducts and therefore more water reabsorption from any fluid intake that you've got. This increase in permeability will allow more water to be retained by the kidneys. The same osmoreceptors will also activate the lateral preoptic area, so an area just behind the the eyes slightly further forward than the paraventricular nucleus and the hypothalamus. And this would stimulate your thirst centre, make you be driven to find water, and increase your drinking behaviour. And all of this would bring the plasma osmolality back to between 290 and 300 milliosmoles per kilo. The opposite regulation is where you've got excessive water intake. And with excessive water intake, you would change the activity of the osmoreceptors. This time you would depress the thirst centre to reduce water intake, and you would also depress the activity of the paraventricular nucleus to make release antidiuretic hormone, which would make the collecting ducts less permeable, so you wouldn't be moving those water channels. The aquaporins would stay in their vesicles, and therefore you would increase water loss, increase fluid loss through increased urine production, and again bring osmolality back to normal. Now, this mechanism we see in excessive water intake in very unusual psychogenic situations, but it also occurs when people take ecstasy and take a range of psychoactive substances, and we see people getting really hot and they drink, but they drink excessively, and they reduce their plasma osmolality to a point that people can pass out, become unconscious simply by over drinking. So it's a big risk for those that take recreational drugs. People go, I know I can get hot, I know that's what ecstasy will do to me, I'll make sure I drink loads. And you see this type of phenomenon that people pass out because they've brought their osmolality so low in that situation. We also see this in marathon runners who are going through water stations, especially you see it in the London Marathon. They're running along, they go, well, I've got to drink at each station, and they overdrink, and they are sweating a lot, losing a lot of sodium chloride on the skin surface as well as water, and they actually end up reducing their plasma osmolality to a point that they need to stop, and they may collapse, and they may be very shaky, jittery, and again, because of the disruption in the plasma osmolality balance, they can no longer perform normal muscle reactions, etc., and have to be hospitalized and their fluid levels adjusted appropriately. Again, giving people very salty foods to try and bring their sodium chloride levels back up. So, overall, we've gone through the different stages of what the kidneys can do. So we've started with the filtration process, and every time the blood flows through the afferent arteriole, through the glomerulus, and out through the efferent arteriole, about a fifth of that blood flow gets filtered, and the filtration fluid that's generated is isotonic, it's the same concentration as plasma. Then in the proximal convoluted tubules, we absorb the major proportion of the valuable nutrients in the tubular fluid. So the majority of reabsorption for any of the factors involved occurs in the proximal convoluted tubule. By varying mechanisms, water, apart from in the collecting ducts, all the water movement will be via osmosis. For other ions, it's a range of symports and antiports, but for other factors such as urea, most of this will be through changes in concentration gradients, so again will be diffusion mechanisms. The osmolality of the tubular fluid that's generated, the time that it starts to be changed is in Lubric Henle with this counter-current mechanism to reabsorb further 20 to 30% of the iron, sodium, potassium, chloride, and further water reabsorption. And then we come down to the distal convoluted tubule, where again we've got some permeability here. We'll get more water movement, more sodium and chloride as well. And then in the collecting ducts, then this is where we've got regulated water reabsorption, but it's only less than 10% of the water that's being reabsorbed across the kidney structure. The final step in the collecting duct is the time that the concentration of the urine will be changed. It will be balanced for the fluid intake levels for that person that time, and so will vary across the day and will vary from day to day, changing not only the osmolality of the urine, but also some of the concentrations that we see, particularly in bicarbonate levels, to regulate the pH of the body, to remove excess hydrogen ions or remove excess bicarbonate if there's been CO2 trapping due to either metabolic or respiratory changes in regulation. So when we look at the overall functions of the kidney, I've got a mnemonic for you to remember this. So when we look at the functions of the kidney, so one of them is acid-base balance, so it's allowing us to keep the pH of the blood regulated by the actions of, as I said, bicarbonate reabsorption from urine and some of the active excretion of hydrogen ions to balance the bicarbonate. We've got the removal of water, again balancing either excessive fluid intake or dehydration, and again that balances the overall urine output. The kidneys are also important in the generation of our red blood cells, and the kidney produces a hormone called erythropoietin. You probably know it more as EPO because it's used as a banned substance and it's used as a performance-enhancing drug to increase red blood cell production to allow greater oxygen carriage within, particularly used by athletes, cyclists, and long distance performance athletes to increase oxygen delivery to muscles. So erythropoietin is produced in the kidney and it stimulates within the bone marrow further red blood cell production. The kidneys are involved in toxin removal, so anytime we take any drugs, the breakdown products of those are filtered in the kidneys and removed because they're actively excreted or secreted into our urine. Blood pressure control is also an important feature of the kidney. Obviously, it's going to balance water load, which can directly change our blood pressure, but it also makes a number of other hormones called renal and angiotensin. Dr. Tunwell will go over those on Wednesday. Now, these hormones particularly balance sodium and potassium and phosphate within the body, which have key roles in regulating our blood pressure further. And overall, we've got electrolyte balances. So we've talked about the different mechanisms that are there, that we can use some of those electrolyte balances, such as the ones in the ascending loop of Henle as loop diuretics to again balance blood pressure, but also change electrolyte balances as well. And finally, the kidney has a role in calcium and phosphate balance by producing active vitamin D. So we make vitamin D all the time in our skin. We need sunlight exposure to activate and start off the first steps in vitamin D production. Then the vitamin D is stored in the kidney where it has further enzymic changes, and then the last activation step occurs in the kidney. So the 1-alpha hydroxylase enzyme is the key, is the last step in activation of vitamin D. And vitamin D is important to lay down calcium and phosphate in our bones. Too little and we will alter our calcium and phosphate balance, which again has key influences on our nervous activity across the body. So overall, we've got a nice mnemonic. What do the kidneys do? A wet bed. So the overall handling of water is key to what the kidneys do, but the mnemonic will remind you of all the different things that the kidney does. So if you remember a wet bed in terms of kidney function, then you can work out all the different things that the kidneys do across the body. Okay, so thank you very much for your attention. Glad we got the slides eventually, and so the last lecture on regulation in the kidney will be on Wednesday morning with Dr. Trumbull. Okay, thank you very much for your attention.